Hi there. In this experiment, we hypothesized that MROB 3014 is an ortholog to B2756. MROB 3014 is a gene in M. ruber, and B2756 is a gene in E. coli. And ortholog is a gene is a gene from different species, or genes that are from different species that are evolved from a common ancestor gene. My name is Samir Abdulkarim, and this experiment was done with the help of Dr. Scott. So what is M. ruber? M. ruber, short for Meothermus ruber. Meothermus means lesser. Thermus means hot, and ruber means red. So this is an organism that lives in hot temperature environment and has a red pigment. Specifically, this is a eubacteria organism. It was first isolated in hot spring in I believe Russia. The optimal temperature for this organism is 60 Celsius. And M. ruber was the study or the subject organism, and E. coli was the model organism for this experiment. We chose M. ruber because besides having the cool red pigment, little is known about its genome, and we chose E. coli because there's extensive published data on E. coli. But before we can investigate our hypothesis, we first have to understand the CRISPR-Cas system. The CRISPR-Cas system is a mechanism of immunity in bacteria. The bacteria use the CRISPR-Cas system to defend itself against bacteriophages and certain viruses. 50% of bacteria contain the CRISPR-Cas system, and 90% of archaea contain the CRISPR-Cas system. This right here, the first figure depicts a picture of the CRISPR-Cas system. This is the CRISPR-Cas array, consisting of the R sequence, which are known as the polygenic repeats, and the S1, S2 through S3 sequence on the rectangles are the spacers. The spacers are pieces of DNA from foreign invaders, while the polygenic repeats are essentially the sequences that are the same from start to finish and finish to start. The CRISPR-Cas system consists of 3 stages: adaptation, biogenesis, and interference. In the adaptation stage, the bacteriophage inserts its genome into the CRISPR-Cas array. Now the bacteriophage genome consists of a sequence known as the protospacer. Within the protospacer, there's a PAM sequence that the CRISPR-Cas system recognizes and then inserts the genome into the CRISPR-Cas array. Once the genome is inserted into the CRISPR-Cas array, it becomes a new spacer. In the biogenesis stage, the CRISPR-Cas array with the new spacer is transcribed and sequenced into a single cRNAs. The cRNAs they target foreign DNA with complementary spacer sequence and presented to effector complexes, which are the Cas protein, to degrade the foreign DNA. The Cas proteins are encoded by Cas genes, which are upstream from the polygenic repeats and the spacers. The CRISPR-Cas system is typically an operon. So this is E. coli's. This is operons for E. coli. As you can see, an operon is an operon. An operon is a unit of genes that are transcribed or controlled by a single promoter. This Cas-AP is the promoter that is transcribing these Cas gene. Specifically, in this experiment, we focus on Cas-E as the gene of interest. We learned that both E. coli and M. ruber contain the CRISPR-Cas system. However, while the CRISPR-Cas system in E. coli is well known, the CRISPR-Cas system in M. ruber is less known. So specifically, we focus on only one of the genes on both the CRISPR-Cas system. On both the E. coli and M. ruber. We focus on we focus on gene known as Cas3 on both of the organisms. So our hypothesis is that MROB 3014 is an ortholog to E. coli E. coli Cas gene B2756. To investigate our hypothesis, we use variety of databases, starting with the EcoScience, which is a database dedicated to E. coli. We use it to learn more about the E. coli CRISPR-Cas system, and then we use Cake to learn more about genes and proteins of the CRISPR-Cas system for both mROB and the B2756, and then we use BLAST to blast B2756 amino acid sequence against the mROB 3014. The pairwise alignment of the DNA and protein sequence determine the degree of similarities. We use IMGM database to confirm that mROB has type 1 CRISPR-Cas systems. So I didn't mention this, but the CRISPR-Cas system are typically categorized into 6 types. Each type consists of a specific signature gene. And so type 1 consists of signature gene known as Cas3. So E. coli is type 1 because it contains Cas3. And then we use TMHMM to predict proteins encoded to have transmembrane alpha helices. We use the ProB, which is this one, the ProB to predict if the protein creates a membrane-bound beta barrel. We didn't emphasize too much on the PSOR because it wasn't working properly, so we didn't really rely on this database. However, we use CDD to analyze both sequence for conserved domains. We use TigerFam to assign proteins to different to protein family based on similarities that the proteins have, and then we use PFAM to analyze amino acid sequence for protein domains, family clans within this database, and then we use the PDB to see if mROB 3014 and B2756 have significant sequence similarity to protein within this highly crowded database, crystallized protein. So this is for the result. This is our first table that we use. This is from the Cake database. So the data displays that both both mROB 3014 and B2756 are assigned the same subunit protein name, which is Cas-E, which is on top right here. Let me move this. And then both of these genes also have similar similar sequence, and their sequence length is also fairly closer. And both of them have the same stir codon, which is again methionine, and even the second amino acid sequence is also the same, and so forth. Okay, next, this is table 2, and this summarizes the result provided from a variety of the databases. I'm gonna put this up here. Okay. So on the first row is the result from BLAST. The BLAST alignment sequence produced low E value of 3e to negative 17, which suggests that mROB 3014 is ortholog to B2756, and a low value typically indicates that the likelihood that the genes are similar is due to common ancestor, not random comparison. The second row shows that is the database is the result from CDD database. According to the CDD database, mROB 3014 and E. coli B2756 code for proteins known as CRISPR-Cas system associated RAMP or RAM superfamily protein Cas6E, and both genes belong to the same superfamily, which is identified by the clan number C108497, and the alignment also provided the alignment sequence also provided a super a low E value, which also indicates low E value, which also indicate that these two genes are similar. And then next we use PrEP. We use the PrEP database and the TMHMM to predict that the protein coded by these genes are localized within the cell. And then the next row shows the result from the TigerFam. The TigerFam indicates that the MROB 3014 and E. coli B2756 have the same protein family because they have the same TigerFam number, which is right here, and they have the same Cas name as well, which is, again, this is the Cas name. They also have a low E value, indicating more similarities as well. From the PDP database, MROB and MROB 3014 and E. coli 27 pull two different PDP codes, but on further analysis, the differences is that the organism from which the protein was crystallized, the 3D protein matched to both M. ruber and E. coli sequence, and then one B2756, again B2756 PDP code for 4DZ, which is 4DZ. Oopsie. Okay. Anyways, one was that B2756 PDP codes for 4DZD, which is the crystallized protein structure for the CRISPR-Cas associated protein Cas6e from E. coli, and then two MROB 3014 PDP code for 3QRPP PDP, which is the crystallized structure of Thermophilus Cas3 bound to an RNA representing a product mimicking the complex. Again, in both cases, they belong to chain A, and the most crucial information from this was that they have a lower E value, again indicating that they're similar. And then finally, from the PFAM, from this bioinformatic database, MROB and E. coli MROB 3014 and B. coli 2756 show strong sequence similarities because they have the same PFAM number, which is right up here. They have the same PFAM. Oops, I'm gonna go back to it again. Same PFAM number, which is right here, and they also have the same name, which is right here. However, the most crucial information from this is also they have a low E value, indicating that these are similar due to evolutionary function. Next, we know that the E. coli contains an operon system. The CRISPR-Cas system in E. coli contains an operon system. We weren't sure if M. ruber does as well. So these are evidence providing indicating that M. ruber does have an operon system. The top is the M. ruber, the bottom is E. coli. The red highlighted genes are Cas E, which is for M. ruber is the MROB 3014, and for E. coli is the B2756. And the neighboring genes that are flanking the highlighted genes are essentially the same on both species. Again, and this is figure 10. Which again, this is a gene neighborhood output showing that MROB 3014 must be part of an operon, which is this is the MROB right. This is the MROB right here, showing that it must be part of an operon. This picture displays that all genes with the same hits in other organisms that are closely related to M. ruber are in an operon. Therefore, it is reasonable for us to claim that M. ruber 3014 is a part of an operon. So, in conclusion, based on the provided data from all the different databases, we conclude that M. ruber's MROB 3014 is indeed orthogonal to E. coli B2756. Both of these are also known as Cas E, and that was it. Thank you.